Welcome to High Strangeness, where we ask questions and explore all that is strange and unexpected in our reality.
Welcome to High Strangeness. I'm Tony, and this is the place where we talk about all things strange and unexpected in our reality. Today's episode, our topic is more of like a did you know in relation to unexpected scientific advances. Now, when I was a young man in junior high and in high school, like the rest of you, I had reports and different papers I had to write, and it was all by hand. And in high school, towards the end, I used a typewriter, but my first time using a computer was actually in college. And I loved it because I could change and edit the work at will. And it was wonderful. But before that, my best friend were things like whiteout and an eraser. Well, what if in life, like let's say healthcare, we could edit problems, mistakes, and mutations? It would change the world, wouldn't it? Well, we're going to be talking about that part of science today and see if we're there yet. Right now, I'm about to talk about a topic called CRISPR. If you don't know what CRISPR is, or if you're wondering where the science is at this moment, this is the episode for you. I've been aware of CRISPR for a few years now, but not so much about how it's progressed recently. So today I'm excited for my guest, who's author Kevin Davies, and his book is Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing. Now, Kevin is a British science journalist and editor. He is currently the executive editor of the CRISPR Journal based in New York. Kevin studied at Oxford University and moved to the U.S. in 1987 after earning his PhD in genetics. He is the founding editor of the journal Nature Genetics and Bio-IT World magazine, former editor-in-chief of Cell Press, and the first publisher of CNN, hope I said that right, the weekly magazine of the American Chemical Society. Editing Humanity is Kevin's fourth book, and he's also was the technical consultant for 2013 movie Decoding Annie Parker, starring Helen Hunt, which I just saw recently and really liked it. It was pretty good. And so with all of that, Kevin, thanks so much for being on the show. Welcome to the show, and how are you doing, my friend?
Hi, Tony. It's great to meet you, and thrilled. Love the subject of your podcast, and CRISPR fits in beautifully to this whole theme.
Yes, it's an amazing subject, and I have to be honest, right up front, a lot of the science is over my head, of course, but the topic is fascinating, and I do understand conceptually like what's happening here. But with the broad range of knowledge of the different people in our audience, I'm going to start off with a few basic questions
Of course,
just to start us off right here.
yeah,
And then maybe after that, we'll get into like breakthroughs, milestones in your book.
yeah,
Sound good?
yeah.
Okay. So let's start off right here. What is CRISPR and how does it function as a gene editing tool?
CRISPR, when it's at home, is a shorthand acronym for a bacterial immune system. And already some of your listeners are probably scratching their head thinking, how the hell does that become a gene editing tool? And it's an amazing story, and I spend the first half of my book telling it, talking about the personalities and the serendipity that is almost always involved in major scientific discoveries. And there were so many good stories about this. How bacteria defend themselves against viruses is a very important aspect of biological research. We have to understand it. Without understanding it, we wouldn't have life on Earth if there were no bacteria. That's how important they are to the ecosystem. And in the 1980s, a Japanese team of researchers were performing some rudimentary sequencing of the DNA of a particular bacterial species. We just recently developed a technology to sequence DNA, the A's, C's, T's, and G's, this four-letter alphabet that all of life shares on the planet. And when they did this in bacteria, they found a weird stretch of the genome of these bacteria, a sort of a strange repeating pattern. It was almost like in England you see this, maybe in America too, in some places. You look on the horizon, you're looking at these sort of verdant green hills, and then you'll see a series of crop circles. You see these sort of strange patterns etched in the landscape, and think, how on earth did they get there? Was that sort of some alien or some farmer getting creative, or what is going on? I think of CRISPR almost as like crop circles for bacteria. There was this pattern of repeated DNA sequences would not have arisen by accident. So there had to be some purpose. And slowly but surely, more and more researchers were sort of, their heads were turned by this, and they started poking around and trying to figure out what was going on. Sandwiched in between these identical repeat chunks of DNA were similarly sized stretches of DNA, about 20 letters long, 30 letters long, of no apparent similarity either to themselves or to anything else that we had sequenced to that point. So it was easy to dismiss them as unimportant. But in the early 2000s, a Spanish researcher named Francisco Mojica was kept running computer searches to try to see if he could identify what one of these intervening sequences what the origin of it might be. And one day he ran that search, and boom, he got an amazing result. That sequence was identical to a stretch of DNA from a virus. And within a year or two, it became apparent that what bacteria are doing, it was as if they were vaccinating themselves with short DNA segments from viruses that they had encountered. And they were using these sort of segments that they were stitching into their own DNA in a way as a sort of an antiviral defense mechanism. So they would then take a stretch of that DNA. We all have heard of RNA now, which is the chemical cousin of DNA. Couple that RNA GPS signal, if you will, to an enzyme that can cut DNA called a Cas enzyme or a Cas nuclease, and then let it loose into the bacterial cell because there's the threat of another virus infecting the cell. And when that nuclease carrying that sort of GPS signal finds the specific match to that signal, boom, the nuclease cuts the DNA and the virus is destroyed. So CRISPR, to get back to your question, Tony, CRISPR is a bacterial immune system. But 10 years after that important, or less than that, 5 or 6 years after that important discovery, the bacteria have evolved CRISPR to defend themselves against viruses. Jennifer Doudna and Emmanuel Charpentier published a major paper, a landmark paper in 2012 that said we can take the elements of this CRISPR bacterial system, put them in a test tube, and if we put any DNA sequence pretty much into that test tube and program the GPS signal I just mentioned to attach to that particular DNA sequence, we can cut the DNA. And the eureka moment was, we haven't perfected this yet, but we now have the basis for a DNA editing system that we can program. We can program just by providing the sequence and literally ordering it online, like we order a book from Amazon or whatever. So now we have a program, a programmable editing system. And another group who published that same a couple of months later, describing much the same sort of system, they coined the term DNA surgery. And when I give talks about CRISPR, that's the one I love to use, because now that hits home. Now we have a technology, it's a nanoscopic technology, or sure, but we have a system now, it's like the ultimate keyhole surgery. But we're not doing surgery on a tissue, part of the liver or part of the brain or part of the spinal cord. It's so much finer than that. We're going into the cell. And now with CRISPR, we can, in principle, edit any particular gene or DNA sequence that we want to edit. And it starts with a cut, but then a whole variety of different biological processes can come in, such that we can now repair the DNA in one of several different ways. So that's the element of CRISPR, and the field has run away with it ever since that 2012 paper was published, because that was the paper that essentially gave Doudna and Charpentier, earned them the Nobel Prize in 2020.
That's amazing. Yes, I remember hearing about it in 2012 and going, what is this? Like, I felt like maybe, and you tell me, this seems like the biggest discovery of mankind almost.
It wasn't clear that it was the biggest discovery when the Doudna-Charpentier paper was published in 2012, but we quickly got the message about 6 months later, because everything I described to that point was doing DNA editing, DNA surgery in a test tube. And remember, these are bacterial systems. The bacteria have evolved over literally hundreds of millions of years. So one of the big questions in terms of, okay, CRISPR sounds great, but is it really going to be able to help patients with different genetic diseases? Well, in order for that to happen, you've got to show that CRISPR can be coaxed into the nucleus of a human cell. The nucleus is where all of our DNA, or the human genome, is packaged in our 23 pairs of chromosomes. And the way DNA is packaged in a human cell is very, very different to the way a single chromosome is coiled in a bacterial cell. There's much more of it, and it's folded and coiled. So could this CRISPR-Cas machine kind of find its way with its GPS signal to the one gene among the 20,000 genes contained in the human genome? Was that really? It was theoretically possible, but could we show it could work in practice? And 6 months after Doudna and Charpentier's landmark paper, several other groups from Harvard and MIT showed, yes, it can work. And so we knew that incredibly that this could work. So within a year after that, all the leading scientists from different universities in California and Europe and Harvard and other places, they all it was like back when you were in school and you would at recess you would pick teams for to play pickup basketball or something, and you'd all line up and wait for the team to be picked. It was like the venture capitalists were doing that. Okay, I'll have Jennifer, you can come and be on my team, and George, you can be on my team. So different sort of dream teams, pickup teams were formed, and so we had companies like Editas and CRISPR Therapeutics, and Intellia was the third one. So these quickly got launched, and then they quickly went public. And from the 2015 to 2020, we entered a sort of a period of CRISPR euphoria, where it seemed like there was just nothing to stop CRISPR from tackling really, any number of different genetic diseases or cancer. And it was that period where CRISPR really burst into the mainstream. It was on every magazine cover. Then 60 Minutes did a program. Then we had Dwayne "The Rock" Johnson in the film Rampage, tackling these humongous creatures that had been morphed by CRISPR reagents falling down from the International Space Station or something ridiculous. Even CRISPR couldn't beat Dwayne "The Rock" Johnson.
Right.
I've watched that film, so you don't have to. Then there was a square on Jeopardy in 2019, and then we had the Nobel Prize in 2020. So way before then, I'd seen how. As I said just before we started recording, Tony, CRISPR wasn't the first mechanism for doing gene editing. In the early 2000s, companies like Sangamo developed a system, and there's one or two other systems as well, but they were very difficult to very specialized, very complicated. You had to pretty much build a whole company to just get one system to sort of work. So there was never any possibility of these really kind of being democratized and being taken up by scientists around the world. The beauty of CRISPR, and the reason we're doing a podcast on it now, is that not just that it's an incredibly successful and powerful gene editing tool, but in any country pretty much in the world, there are scientists using CRISPR. It's so easy to work. You can order the reagents you need online. You don't need fancy pieces of equipment or, billion-dollar research institutes. There are literally high schools around the country and beyond that are teaching seniors. It may be in advanced biology, but there's still it's that level that we're at now, that you can do a rudimentary CRISPR experiment. And I'm sure once you do one, you think, oh my God, this is so cool. How could I, not want to play with this in my career?
Yeah, that's amazing. Two things came to my mind as we're talking and as you're explaining some of this. One is that a couple 100 years ago, without us even seeing and knowing much about bacteria, who would know that the answer to so much of potentially our future fixes, right?
Yeah.
Come from that little microbe,
It's so funny, because when I was doing my PhD in the 1980s in London, we were the big tool that we would use every day in the lab was a series of enzymes from bacteria. So the first wave of genetic engineering that really gave rise to companies like Genentech and Biogen and Genzyme, the first biotech companies, this all came about from the discovery in bacteria of so-called restriction enzymes that could cut DNA in very specific spots. And this was such an amazingly useful tool, because it allowed us to prise open a bacterial gene or chromosome, insert a human gene, and then use the bacteria to grow up the gene and produce the corresponding protein. And restriction enzymes are part of an earlier, previously discovered bacterial immune system. So we learned in university, at least I did, about bacteria have this immune system. It's marshaled by these enzymes, and that one's in every textbook. And we thought that was it. And it's crazy how biology, there's still so much amazingly cool biology that we don't understand, and we've had to rewrite all the textbooks again, because CRISPR was it was sitting there in plain sight, but we only began to get the gist of it in 2010, 2012, and other forms of bacterial immune systems have been discovered since then, which may or may not have gene editing applications down the road. So it's why biology is so cool.
It is so cool. And it was my favorite subject
in
college and in high school. It really was. I was just amazed by it.
Yeah.
So before we go any further, I'd like to just go a little more granular about one thing. The components, you kind of went into it, but the components, subcomponents of the CRISPR system.
Yeah.
Like one of the things I've heard is Cas9, 12, 13.
Yes.
And I don't really think I understand what that means. It's a protein. Could you talk about that?
Sure. They're just different flavors of CRISPR, so that they will cut DNA in slightly different ways with different sort of specificities. But the first one, Cas9, is the one that is in the clinic now. And there are some trials using some of the other Cas nucleases, but your audience doesn't need to worry about it, because they're all essentially acting in the same way. So when we talk about CRISPRing a gene or a patient, really all we're doing is we're giving them two things: the enzyme that is the genetic scissors, as the Nobel Prize Committee called it when they gave Doudna and Charpentier the prize. So that's the thing that actually does the nicking or the slicing of DNA. And then we combine that with an RNA, and we have synthesized, in the computer, we've typed out the sequence that matches the gene that we're looking to target. And there are other parameters and other factors that we have to consider. It's not, I can't claim that we can edit literally any stretch of DNA. One thing that we have to look for in doing a human experiment is that we want to design this so-called guide RNA to focus and align with the gene that we're looking to edit. But hopefully there are no other stretches in the genome on some other chromosome, the other side of the dark side of the nucleus, where that same 20-letter stretch of ACC, TCC, AC, whatever it might be, has also exists in some other gene or some other chromosome just by chance. If you do the back of the envelope math, you'll come to the conclusion that for any 20-letter stretch that you're trying to target in a specific disease gene, there is a finite chance, it may be 0.0 something percent, but there might be a slim chance that that same sequence exists somewhere else. And the CRISPR enzyme that you're delivering into the nucleus isn't going to know that, well, I'm allowed to cut the one on chromosome 6, but I'm not allowed to cut the one on chromosome 13. It's going to see that sequence, it's going to cut. So part of the game and part of the early safety concerns around CRISPR was these so-called off-target effects. How can we increase the signal to noise? We want to keep the noise down as low as we possibly can. And for the most part, it seems that the scientists have done that very successfully. And when the first CRISPR therapy that was approved 12 months ago, Casgevy for sickle cell disease, when that was approved, the FDA held a hearing with the company that was driving this clinical trial, Vertex Pharmaceuticals in Boston, a big FDA hearing with a 15-person blue ribbon panel appearing by Zoom of experts to help the FDA, to give the FDA some certainty that the sponsoring company had done everything in their power to ensure that the therapy, that the editing was safe and totally specific on the gene that they were trying to edit, and there were no safety effects. And they passed that sort of exam with flying colors. So, and the results can be seen in the many patients. And we'll come to that later on, I hope.
Yeah, and that was one of the questions I was going to ask later about the off-target possibilities. So it sounds like that's part of the vetting before they can say we're ready to go.
Very much so.
Making sure.
Yeah, very much so. Very important part. Yeah.
Perfect. Okay, I do have other questions, but I think we may be able to answer them through the discussion of. I'd like to go through maybe points in history and the breakthroughs, which you have covered in your book.
Yeah.
Maybe we can weave in and out of those breakthroughs, milestones, and your book.
Yeah.
That would be awesome. And I know in 1987 was the first time some things started to happen. Could you talk about that for a minute? And then I know there's stuff between then and 2000.
Yeah.
Maybe give us a little.
Yeah, sure. So I mentioned the Japanese team that first reported on these CRISPR repeats. That was in 1987. Very little happened in the 1990s, but the most important thing was that this Spanish microbiologist, Francisco Mojica, who was really the first person I went to visit when I started reporting for the book, partly because he lives in a beautiful part of the world, Alicante, on the east coast of Spain. But I was fascinated by what had drawn him to this story. About 10 miles south of him is a fascinating place, Salterns or Salt Lakes. It looks like you've sort of stumbled into a David Attenborough, planet Earth. It's such all these beautiful lakes. There are flamingos and a stench of sort of sulfurous bacteria in the air. But this is a working mine where you are literally, the company is literally crystallizing salt out of the Mediterranean. So there are these big lagoons of increasing salinity as the water gets more and more concentrated with salt. Yet there are bacteria and other microbial species that live really happily in these incredibly harsh environments. And Mojica wanted to understand what about what is encoded in their DNA that helps them survive this. These are such salty conditions. You would think that by osmosis, the salt would just be sucking all of the liquid out of the bacterial cell until it was shriveled into nothing. So he thought, his original theory was that these funny CRISPR repeats that the Japanese team had published on in the late 1980s, maybe that was the key. There was something going on there. And when I give talks, I say it was a lovely theory. It was completely wrong, but it kept him hooked on what CRISPR was. And then, of course, as I said, in 2002, he made this dramatic discovery that the CRISPR array was housing snippets of viruses. He published that work in 2005, and then, as did two other teams, about more or less at about the same time. But that was all work done in the computer. The big question then was, okay, it looks like CRISPR is an immune system, but who's going to be the first team to prove it? And that work was led by a French scientist working in America named Rodolphe Barangou, with whom I now work very closely. He's the editor-in-chief of the CRISPR Journal, which is a journal I sort of conceived of and launched about 7 years ago with the Marianne Liebert publishing company in New York, who you referenced in the introduction. So Rodolphe was leading a team at a yogurt company, of all places, and he and his team were able to show that by changing the CRISPR repeats that they encoded in the bacteria that they were using as starter to start dairy cultures, that changing the CRISPR sequences changed the defense profile against different viruses. So they could correspond. If you put in the sequence that corresponds to virus X, that culture was resistant to virus X because of the mechanism that we discussed a few minutes ago. So by 2007, that was published in Science, the very high-profile, very prestigious journal. By 2007, there was no doubt that CRISPR, what the function of CRISPR was, and then more and more scientists gravitated to this. This was interesting. There's some new biology here. There are some new enzymes here. We want to understand how they work, and we really want to understand at a deep sort of molecular, almost the atomic level, how does CRISPR identify and then target these viral DNAs. And we talked then earlier in our discussion about the Doudna-Charpentier landmark paper in 2012. Doudna and Charpentier only met a year before. They were both attending and speaking at a conference in Puerto Rico. And Emmanuelle was the sort of junior partner of the team. She went up to Jennifer and she introduced herself and said, I'd love us to work together. And they decided to go on a walk through the cobblestone streets of old San Juan to just talk about what their different areas of expertise and what a collaboration might look like. And I guess by the end of that walk, they said, right, let's work together. And so they don't collaborate much of at all anymore. But that 12 months or 15 months that they worked together, I mean, chef's kiss. You could not dream a better collaboration. They then went on to work on, I mentioned the schoolyard and the picking the teams. They ended up on different teams. And that was kind of interesting. So Emmanuelle didn't really have the first clue about how to start a company, but she had friends who, from her postdoc days in New York, who had gone on to take big executive positions at pharma companies. So she called one of them, and he and his advisors and colleagues immediately saw the potential. So they said, Emmanuelle, oh my God, there's a company here. And so that eventually became known as CRISPR Therapeutics. Jennifer was drawn to and a co-founder of Editas, which really had most of the rock stars in the field, George Church, David Liu, Feng Zhang. Some of your listeners will recognize these names. But then something very interesting happened in the annals of CRISPR. Again, I devote a whole chapter in the book, Editing Humanity, to this. This was a patent battle suddenly erupted out of nowhere Feng Zhang had been one of the scientists who had shown that CRISPR could work in human cells, which was a very important discovery. And indeed, I think for some, perhaps you could make an argument that that would have could have elevated him to winning a share of the Nobel Prize, because without being able to get this to work in humans, really the interest commercially and scientifically would be much lower. But Feng was not the only scientist to make that statement discovery in early 2013. George Church, famous scientist at Harvard Medical School, published essentially the same result in the same issue. So now you had the two women scientists who made the first important discovery. Now two former colleagues, Zhang and Church, had shown conclusively that CRISPR could work in human cells. But even if the Nobel Committee said, yeah, both of those, let's wrap those together in a prize, the Nobel Committee only accepts 3. And there was no way you could break up Zhang or Church. You couldn't pick one of those. That would be totally unfair. So I think that's in part why we had this clean break. But in terms of the patent for CRISPR, that played out a little differently. Zhang decided, he with his advisors at the Broad Institute in Cambridge, Mass., to apply for a patent, and they were able to leapfrog ahead of Jennifer's own patent application. So now suddenly the two scientists who were co-founders of the same company were now squaring off in the patent court. Jennifer decided this was unsustainable, so she resigned from the founding team of Veritas, and then went off to launch a number of other companies. But the way that patent has played out is that Zhang did eventually, after years and years of wrangling, at least in the US, receive the nod from the patent court. We would need many episodes of the podcast to really do that story justice. But I can give you a guest who you should have on if you want to follow that. So when Vertex won approval for Casgevy for sickle cell, before they could commercially develop that therapy, they had to pay a big, tens of millions of dollars, I believe, to Editas for the rights to commercially use CRISPR-Cas9, because those rights had been that's where those rights lay. So that's where we are.
Amazing. Well, and I just want to say this thing to the audience. We're kind of skimming on information here, but the book, which I read as well, Editing Humanity, wonderful. You don't have to be like, I love the way you put it in such a way that a broad audience could understand what they're reading, which is great, because I've mentioned before, I have some understanding, college level biology and stuff, but that's pretty much it. And so it was very easy for me to follow. I knew there were certain things over my head, but great book, great information, lots of details. So people, you need to pick this book up, Editing Humanity. Too bad this is coming out after Christmas, but there are birthdays, so make sure you get
yes
for a birthday present.
Right. Yeah. What he says. Yeah.
Yes. Absolutely. Okay, I'm just looking through some notes here that I have as well, and I mean, this was a big time in that 2012-13 area. Yeah,
and since then, what's been exciting is that newer. The world didn't stop there. We didn't say, okay, we've got a gene editing system. We can now go on and do something else. Many of the scientists we've been talking about have continued to develop new flavors, new forms of gene editing. So some of your listeners will have heard of perhaps base editing or prime editing. There's a researcher in Boston, a colleague of Feng Zhang's named David Liu, who, with the assistance of a string of incredibly talented postdoc scientists who spend a couple of years usually in his lab, have developed even more sophisticated versions of gene editing, where we are now literally performing chemistry on the double helix to sort of excavate. It's almost like repairing a pothole, where it's sort of taking out one of the letters and doing some pretty clever, nifty chemistry so that we can now lay down a different letter or nucleotide to make a single letter change in the genetic code, which is 3 billion letters in the human genome. So this is incredibly sophisticated. It doesn't you can't get any more precise than just making a single letter change in the human genome. So both of those companies are now embarking on clinical trials for different genetic diseases. So that's exciting. And then also just, I think, about to get underway with a clinical trial, we have the first in at least a couple of companies doing epigenetic editing. So rather than changing the raw sequence of our DNA, it's about changing the chemical signals and motifs that are layered on top of the double helix that change the way that different genes are expressed. So the idea is that we can we can perhaps correct a specific disease, not by interfering with the actual sequence of the DNA, but by changing the way that different chemical groups are attached or removed from that gene sequence to either suppress it, shut it down, put it in mothballs, or kind of give it the kiss of life and let it reawaken. And it's the latter, that has really been the key to the success with the sickle cell disease program. Rather than trying to change the single mutation that causes sickle cell disease, researchers have said, let's reactivate a form of hemoglobin that we all express before we're born. So it's literally called fetal hemoglobin. The key gene behind that molecule is in our genome, but it's been shut down. It's like we put a wheel clamp on it in the months after we're born, and a different gene is switched on. But the different gene that is switched on and forms half of our hemoglobin, which is the protein that carries oxygen around our bodies and keeps us alive, that gene that is switched on after birth is, if you are unlucky enough to have sickle cell disease, that's the gene that has the mutation. So it's an incredibly ingenious workaround. It's like taking the spare tire out of the trunk of the car and sticking it on the hemoglobin. And based on the results in the first many dozens of patients who've been treated since this trial got underway in 2019, it works better. It works beyond anybody's wildest dreams. And so that's the poster child for the clinical possibilities of CRISPR and gene editing right now.
Yeah. My gosh, the advancement in such a short period of time in this area is exciting, amazing, but like scary. Like it's going so fast.
Well, it's funny that you say that, because for the people insiders in the field, it is not nearly going fast enough.
Really?
So what do I mean by that? It's and of course, we're entering a new presidential administration where, based on the names that are being floated and maybe even be appointed by the time this episode airs, we may see wholesale changes in the health department, Health and Human Services, which of course oversees the Food and Drug Administration, which regulates all of these drug therapies and these newer gene therapies, including gene editing. And a scientist who works with Jennifer Doudna very closely, Fyodor Urnov, one of the pioneers of gene editing, he's been in this field for 25 years. Fyodor has published numerous articles and given many talks around the world, raising the alarm and saying it's really not the FDA's fault, because they have to establish routines and pipelines and processes. Drugs, before drugs are approved, they need to be vetted. I think we can all agree on that. And so the question is how rigid and how when do these regulations just become a bureaucratic mess, and we need to kind of unravel some of them and think a bit more logically about it.
Yeah.
And Fyodor has been making the claim that at the moment, every time we design one of these new RNAs, the GPS signal, to target a new mutation for a new genetic disease, the FDA has to treat that as a new drug. And so we have to start at square one and do all the preclinical and all the animal model studies. And even if that gets approved, if the same company, the same scientists say, right, we now want to turn our attention to the second most common mutation in that same gene, it's like Chutes and Ladders. You're that close to passing, winning the game, and you've got to go all the way back to the beginning. And Theodore and others are saying the FDA, and I think there's a lot of sympathy from FDA executives, administrators, that yes, we have to figure out without compromising patient safety, are there areas where we can say, okay, you did it over here. It's just like changing the topping on the pizza. Just because you've changed from pepperoni to green peppers and mushrooms doesn't mean we have to now qualify the chef to show us how to roll, right. bake the dough. We understand that hasn't changed.
Yes.
So Theodore published an op-ed in the New York Times in the late 2022 or 2022 that people can look up, and has just published a masterful 6,000-word editorial in the October issue of the CRISPR Journal, which I urge people to look up. That gives us his latest thinking, and not just that; it's a recipe for how we can break through. And so, to distill all that down, you said we're going too fast, but we've only got one CRISPR therapy approved by the end of 2024. Yeah. We may get a couple more in 2025, but this is snail's pace given how many thousands of genetic diseases there are. And the scientists like Jennifer Doudna, every week they are receiving emails from the families, the parents of patients with a terrible genetic disease saying, I've heard about CRISPR. We've tried everything under the sun. We think CRISPR is probably the only tool that can help us. How can we enroll in a clinical trial? And it brings these scientists to tears, because practically there's very little they can do, but theoretically they know that 2 weeks from they could order the reagent that could in principle cure this child if only we had the ecosystem. And that may be increasingly turning to the universities, turning to nonprofits to do this for these very rare diseases. A Pfizer or a Sanofi or a big pharma company is really not going to have much interest in tackling a genetic disease where there's only 10, 20, 30 or 200 patients. Occasionally it can work. It can be economically profitable for them. But, unfortunately, it's got to be economically viable for them to do this. With sickle cell, none of the companies that were involved in getting Casgevy approved, they certainly haven't gotten their money back. They're charging $2.2 million for this therapy, which for many will seem like an outrage, but it's actually only half the price of some of the cell therapies that have been approved in the last couple of years. But there is a patient pool of perhaps 100,000 sickle cell patients in the United States, millions more, of course, in Africa and India and Asia. But of that 100,000, there may be perhaps 20,000 who are on the more severely ill category, who nevertheless are not so damaged with organ damage that they wouldn't qualify for the trial. So that's for a rare quote unquote disease. Sickle cell is a very common disease, and this is a patient population that has been yearning for something to cling to, any kind of hope, for decades and decades. It was first described in 1910, for goodness sake, and the only real medication they've been offered was approved in the 1990s, called hydroxyurea, which is a sort of an ex-chemotherapy drug, but it helps to turn on this fetal hemoglobin, so it does ameliorate some of the symptoms and pain crises that these patients experience. So to go from Doudna Charpentier publishing in 2012 to the Nobel Prize in 2020, that's really rapid. That's amazing. But now we're seeing the roadblocks in terms of getting this therapy into the clinic, and so the clarion call has been sounded, and it'll be interesting to see if, based on who we think may be leading HHS and
Yeah.
FDA, there's clearly going to be some shakeups, and maybe this will actually help for this
Yeah.
in this particular context.
Yeah, bottlenecks, regulatory bottlenecks, and why not bridge work that's common, right? That's what
Yeah,
we're saying.
yeah,
That's the key. There was the COVID stuff that
yep.
they fast paced. So I wonder if there's an appetite to fast pace this kind of stuff.
Yes, I think that's a great analogy. Operation
Responsibly.
Warp Drive you're referring to, which,
Yeah.
yes, so we got those first vaccines approved in less than a year. And so that was great. And so I think people feel the urgency, because there are something like 5 to 7,000 known genetic diseases where we know the gene and the mutation or mutations within that gene that give rise to this particular genetic disease. And in principle, CRISPR could treat and perhaps cure many, if not most of them.
Yeah.
So that's the upside. That's the glass is half full.
Yeah.
And if we can figure out a path through some of these regulatory bottlenecks, which exist, I think, for good reasons. This is not to malign the FDA. They have to establish processes. But sometimes you end up putting a square peg in a round hole. And when these processes were laid down, gene editing was not a thing.
Yeah.
And now we see the flexibility and the programmability of CRISPR gene editing and base editing and prime editing. And the FDA now is on high alert that they have to adapt the system in order to change with the times.
Yes. Yes. No, that's amazing. And I was going to ask you a little bit about the regulatory environment. So you've gone there. Thank you. Because I mean, I almost wonder if we don't get this right, it will force people to leave the United States, like they've had to do for other things, for treatments. I'm assuming this is available other places, or
well, except we touched on the high price. So most of the thought leadership is in the United States. There's absolutely no question about that. And most of the commercial development, if not all of the commercial development, pretty much is happening here. So despite the red tape, if we want to call it that, that currently exists, this is the place where the financing exists. This is the place where the companies and the thought leadership, for the most part, exist, and where the trials will kick off. That doesn't mean that the trials won't get done in New Zealand or Europe or places, but the inception of these therapies is less likely to happen in Europe, for example, because they have different tolerance for pricing. Here, when a company gets a drug approved, they can set their price. There are advisory committees that recommend what those price thresholds should be, but ultimately it's up. The government doesn't tell them what to do. But if you then try to sell that drug in Europe, whether it's a drug or a gene therapy, you come up against the government and committees that are run by the government. Because in Great Britain, for example, the National Health Service cannot just bend over and pay whatever Vertex or Biogen or whatever the company is, whatever they demand. So there is a negotiation, and the US companies know that they're going to be bargained down on the price they would like to set, and they do set in the US. And the question is, can we find a compromise? And those negotiations can drag on. They can be very tough. And US companies have pulled drugs from Europe because they could not make them economically successful. And that may change 10 or 20 years from now, when there are multiple companies offering different versions of gene editing for hundreds of different genetic diseases, and we get much more competition and the therapy is more accepted. Then maybe we'll see it more widespread. But so that's a challenge. The other challenge, as far as the sickle cell therapy, which has been approved, that's fantastic news. And I can tell you about some of the patients I've been interviewing for that. But the problem there is that it's a very it's a torturous process. We have to harvest the stem cells from the bone marrow from the patient. Then we treat them in a lab outside of the hospital with CRISPR to fix the gene in question, and then we collect them and concentrate them, and then months later we give them back to the patient, but only after the patient has undergone chemotherapy, which is brutal,
Yes.
to provide room inside their bones, in the bone marrow, for these new stem cells to find a perch, literally, and take root. Where then, for the rest of the life of that patient, if all goes well, they will keep pumping out and developing red blood cells and all the other blood
Yes.
cells that the patient needs. Um, this there are, obviously this is the facilities needed to do this are widespread in the US and in Europe, but really do not exist for the most part in Africa, where most of the patients are. So on the one hand, we're super excited about this milestone of the Casgevy approval because the clinical results have been spectacular. But no one is under any illusion that this is going to work in Africa, not just because of the price that is being quoted, but because the facilities just don't exist there. So
Yeah.
even the company Vertex that won approval for Casgevy, it has already said that, as thrilled as they are, and obviously they're going to do their best to make this a commercial success for the company, they also have a different team of scientists trying to use the information that they've learned from the biology of fixing this gene to now, can we make a small molecule, a regular aspirin type pill that patients would take on a daily basis that can do what the gene editing does? Because if they succeed in that, now they've got a drug that they can sell around the world,
Yeah.
and that will be an amazing day.
Well, that's a question I was gonna ask you was what does treatment look like? Is it a dose here, a dose there? Is it like chemotherapy and that's interesting to me. I was curious about that though. Is it different? Like in sickle cell, you have blood, but if you have some other genetic disorder, is that different than what you would?
The process will differ depending on which disease you're talking about. So for
Yeah.
sickle cell disease, but in principle, the gene, if we're editing genes in blood or the liver or wherever, we only need to do that once. So that's another reason that CRISPR is so exciting. This is not a therapy where you've got to go back every 3 months or once a year to get a top up. On the contrary, so yes, the sickle cell regimen for the patients was brutal, and not everyone was fit enough to do it,
Yeah.
and many were scared to even entertain it. Um, but you only have to do it once, because a
Wow.
stem cell, by definition, is a cell that for life will keep developing, and the more specialized cells, including the red blood cells that do the oxygen transport, which have a natural half life in the body of about 4 months. So they're whizzing around our body as we speak, trillions of them, literally,
Yeah.
and it's a punishing it's a punishing life. And after 3 or 4 months, they wear out and they are replaced by our bone marrow is making millions of these, every second as we sit here talking. So, incredible, isn't it?
Well,
But if you were treating a liver disease, you would go a different route. You would be So the next therapies that we think the FDA will approve, perhaps in the next 12 months, are from a company called Intellia, which has had equally exciting clinical progress, treating a pair of lesser known liver diseases where we're not going to take the liver out of the body and then transplant it back. That would be crazy.
yeah.
But scientists have developed other vehicles that act like a Trojan horse that are able to smuggle the CRISPR machinery into the liver cells safely, where they can make their fix, and then they're sort of turned over and the CRISPR disappears. And these liver cells are fixed, and now the results in most of the patients treated for a couple of different liver diseases are looking really, really, really good. All these results are all published in the New England Journal of Medicine, which is the elite medical journal. And as we think about tackling muscle diseases or hearing loss or diseases of the eye, we know we have strategies to go after those specific parts of the body, those specific tissues, and we can do that. We can do that very safely. So, yeah.
That is fascinating and exciting. As somebody who just got done 3 years of chemo and radiation therapy myself,
Ah,
to have an opportunity in the future for people to
yeah,
have to not go through that,
yeah,
wonderful.
yeah, yeah. So that's great.
That's incredible. Okay, this is great. I knew we'd get to some of the questions I had. The one thing I did want to talk about, though, well, two, real quick, is the application in agriculture.
Yeah.
Do we see CRISPR playing a huge part in this area or this space?
I think we do, and I think we need to. So I devoted, I think, a couple of chapters towards the end of the book about the fact that, as everyone knows, the climate is changing, and pretty much all the foods and crops and plants that we eat are under threat from something. It might be drought in one part of the world, it might be some bacterial pest, it might be a parasite. And if we just let nature play its course, we're going to lose the ability. We're going to lose oranges, we're going to lose
Yeah,
bananas, we're going to lose many of the foods and fruits and vegetables that we rely on. So CRISPR is a safe and a powerful way that we can genetically tweak the DNA of these species that we want to eat to give them resistance to all of these threats and many others. Um, of course, the public will look a little skeptically. Many of them will, because, of course, we've all been uh, sort of brainwashed in a way by the threat of genetically modified foods and organisms, and they sound inherently evil and dangerous. I have a different opinion that but even if I were to say, okay, I understand your concerns. You don't like the idea of taking a gene from, say, an insect and putting it in a plant. I would counter that by saying, forget where the gene came from. It's an inert
right.
chemical that we are stitching into another inert chemical. There really is nothing to be nervous about. Anyway, but let's say no, people have concerns. With CRISPR, that's not what we're doing. We're literally taking this molecular scalpel. You made your analogy in the introduction, and going into the genome of the plant and making the most subtle fix. And the irony is that now, so that's the scientifically savvy way of engineering the future of our food supply.
Yeah,
The other way that is currently legal is to just take plants and vegetables and bombard them with X-rays and mutagens to sort of fast forward the evolutionary process in the hope that one of these DNA variants could spontaneously arise, and then we can cross it back with our root stock and eventually get to the plant, get to where we're trying to go. But that could take generations. And with
yeah.
CRISPR, we can now do it, perhaps in a period of a few months. So
With
why
CRISPR
wouldn't
being
we?
precise
Yeah,
versus this bombarding
exactly.
of
Versus
mutations.
sledgehammer, brute force,
Yeah,
trial and error approach. So the battle there now is with the regulatory agencies to provide enough evidence and common sense to say there is no inherent danger, because all we're taking, it's the same. If we were to do this in bananas, for example, it's the same species. We've just made the most subtle genetic change imaginable. We are not introducing something alien. This is
right.
inherently safe. And obviously, we'll do the field trials and tests and taste tests, and but so this is so this has to become important for agriculture. And don't take my word for it. If Jennifer Doudna was the Nobel laureate, was your guest, she would say, and has said many times on the record, she, for all the excitement around treating patients with sickle or other diseases, in the long run, she thinks the application of CRISPR in agriculture is going to be the most profound impact for this technology.
That makes sense. Okay, so I've got one last CRISPR two-part question.
Yeah.
And to me, it's the big one that probably people eventually get to, which is risk.
Yeah.
So there is a podcast, Patrick Bet David Valuetainment podcast, where he brings up something. He took about 10-minute section to talk about CRISPR. And he talks about all the good and bad, but he does it quickly in 10 minutes. One of the things he does say is that in 2020, a U.S. intelligence report showed that China conducted human testing on members of the People's Liberation Army to develop soldiers biologically enhanced. And it's funny because early on when I heard about CRISPR, the first thing I thought is, in 25 years, we're going to run across a real X-Men who escaped from a CIA underground thing because they can't help but to do this, right? Because now that it's available. So my question to you is, what are your thoughts about that? Like, this is definitely something we need to do, but the technology is now out there, and there are the bad actors, of course. So what do we say about this? Are we going to see something strange down the road? And I'm curious what's the extreme, or what's the maximum change a human could see?
Yeah.
Will they have wings? Who knows?
Yeah. Yeah, I think that would be strange, and I think that's a little too far in the future for any of us to be too concerned about. I'm glad you brought up. So I have no further information to supply about X-Men and countries performing potential experiments on developing the future military. I did have a quote in the book, though, from Putin, who was speaking to his students 5 years ago in a public forum on the record, also sounding very enamored by this possibility. So clearly this is being discussed. Your question is a good, really segue to something I really want to spend a bit of time talking about, which is that to engineer somebody, a new generation of something, you would have to make a DNA change, a heritable DNA change, which means performing gene editing in the embryo. So all of the examples we've talked about before, up to this point, Tony, in the program, there's been no ethical controversy. If I treat the blood of a sickle cell patient or the liver of a patient with a liver disease or somebody who's blind, there is no chance whatsoever that those CRISPR edits could somehow magically become incorporated into the egg cell if in a woman or sperm in a man and then be transmitted. So these are so-called somatic changes rather than germline changes. Yeah. But if I do a gene editing experiment on a human embryo, a single cell human embryo, and it's successful, then as that embryo divides into 2 cells and 4, 8, 16, and so on, every cell from that original single cell will carry the edits that I've introduced. And that means that those edits will be present in the egg cells and the sperm cells, or half of them, that are then poised to be transmitted, and passed along to the next generation. And against all odds and against all conventional rules, laws, and wisdom, 6 years ago, almost to the day that we're recording, a Chinese scientist took this path. So He Jiankui was a young Chinese scientist who, after his biology degree in China, came to America, did his PhD at Rice University in Texas, very good school, then did a 1-year postdoc with Stephen Quake at Stanford. Steve Quake is now the co-director of the Chan Zuckerberg Research Institute. So He Jiankui, I'm going to call him JK for short. JK was rubbing shoulders with some very smart scientists. And then he got lured back after 1 year at Stanford to launch his own group the Chinese shower their star students with funding to come back and build their own research teams back on the mainland. So he launched a group in Shenzhen, China. Shenzhen is the massive city just across the border from Hong Kong. And he, for the first few years in his lab, JK had success. He built a DNA sequencing company. He was making money. He was certainly drawing attention, having success, publishing papers. But we talked earlier, didn't we, about how easy CRISPR was and high school students can do it. And somewhere along the way, JK decided he wanted to make a name for himself. And CRISPR was the way to do it. And why just limit yourself to treating patients? Why not do the ultimate experiment and CRISPR a human embryo? But not just for research. Why not go a step further, that big step further, and implant those gene-edited embryos into patients, into a woman to start a family? What would be a good medical excuse to do that controversial experiment? So he found an excuse. HIV is still a big problem in parts of China. And there's a gene we know that if it carries a mutation, which occurs naturally in a small percentage of the human population, the gene is called CCR5. If we delete a section of that, CCR5 encodes the lock to the HIV key. And without this lock, HIV can't enter the cell. So if we gum up the lock, HIV's got nowhere to go. So he decided he would try to use CRISPR to replicate this 32-letter deletion in the gene, the CCR5 gene, that millions of humans have naturally and are HIV resistant. And so he went about that experiment. He got a very dubious sort of ethical remit from his university. He told nobody that he was doing this, because if he'd announced his plans publicly, he would have been vilified. Yeah. So he said, you know what, I'll deal with the flack afterwards. He informed a few confidants in confidence that he was doing it, including Quake, including a couple of bioethicists at Stanford. But they, interestingly, they were sworn to secrecy, so they even though they didn't agree with what he was doing necessarily, they decided, it would be wrong for me to abandon that. Plus, they didn't know who to call. Who do you call? Right. The World Health Organization, the director of the NIH, the White House, the New York Times. Nobody knew quite what to do with this explosive information that now, in early 2018, a woman was pregnant, carrying twins. And those twins were born in either late October or early November of 2018. And at the end of that month, Thanksgiving, it was Thanksgiving week, or the week after Thanksgiving, there was a conference, a bioethics conference in Hong Kong, and I made an excuse to go and attend that conference, thinking it'd be interesting, but not really expecting any fireworks. And holy cow, JK was scheduled to speak. The news of the twins being born came out thanks to some brilliant investigative reporting by a journalist at MIT Technology Review named Antonio Regalado. He'd met JK a month earlier in China while filming a documentary. It was a strictly off-the-record meeting, but Antonio came away from that meeting thinking, this guy he's clearly poised for all the work he's been doing in monkeys. He's poised to go the next step, and I get the feeling he wants to, and he probably already has. And then Antonio found a record on a Chinese website that convinced him that experiment was underway. And so he published his hunch. JK didn't deny it. JK's PR handler didn't deny it. In fact, begged Antonio to hold off on the story, and they would try to give him some exclusive information later on. JK said, no, this is the scoop of my career, potentially. So he published the story, Thanksgiving Sunday, and it was right. JK immediately uploaded some videos on YouTube announcing the birth of Lulu and Nana. And then 72 hours later, showed up at this Hong Kong conference. I was in the front row. There were hundreds of journalists lined up. It was like a rock concert rather than a bioethics symposium. It was surreal. And he tried to defend his experiments. He faced a battery of hostile questions from the audience. He didn't have good answers. His paper was never published. He was immediately taken into house arrest because the world's press, every expert being interviewed said he was wrong to do this. And it wasn't just a matter of, well, that's a bad experiment. You're playing with the lives of human beings. What gave you the right to do that without consulting all the other key stakeholders of your species? Not just handpicking a few people to then swear to secrecy. So JK served 3 years in jail, close to, for breaking various sort of laws. And but interestingly, now he's been released a couple of years ago, and he's been rehabilitated. So if you can look him up on X, formerly Twitter, he's got a new lab in Beijing, and he's emboldened, it seems to me. He's talking more openly about repeating these experiments. This is the future, he says. We should do this for Alzheimer's disease. Nothing about the military or making them strong or improving their vision or speed or whatever. So I've got a feeling we're going to be going down this same road again at some point. Unfortunately, as for the military, just to humor you for that question, what DNA would you edit? I mean, is this to give them a bit more muscle? I mean, can't you just go to the gym for that? What exactly? Yeah, and for many of the traits, often this comes up. Well, can't we engineer embryos to do better on their math tests or improve their intelligence or their musical prowess? Not really for the military, but for other sort of dystopian sort of notions. In many cases, my pushback would be you haven't got a clue which genes would be involved. Probably because there are so many that contribute to something like intelligence. You can't just say, well, if I tweak this gene and that gene and that gene, I will earn my future child 5 extra IQ points. Really? What is the point? You're dreaming, you're hallucinating. So I'm not too worried. You mentioned bioterrorism, though, bioweapons. Often people worry about CRISPR, and there could be labs. There's still debate, isn't there, raging about COVID, the origins of COVID. Was that produced in a lab? Could CRISPR develop a more potent bioweapon? Well, look at the number of lives that COVID took. This look at still the outbreaks of Ebola and other natural microbes. I really think worrying about some CRISPR lab engineering it. Nature does a perfectly good job of keeping us on our toes and worrying about the next pandemic, which could be about to hit. That's a bit of a red herring to me.
Yeah. Well, I just couldn't help it because I know some of our black projects, they can't help it. They'll just push every button they can, and who knows.
Well, it's the High Strangeness podcast after all, so
we got to think about it.
Blocking the question at all. I expected it.
Awesome. Well, look, this has been great. So, just as we close this up here, the progress, the next 5 to 10 years of CRISPR.
Yeah.
What do you see? Providing we can cut away some of the bottlenecks.
Yeah.
What do you think?
More clinical trials. Certainly hoping that CRISPR remains safe. Just in early November, one of the CRISPR or gene editing companies called Beam Therapeutics announced the first results on half a dozen patients in its own sickle cell clinical trial. And the results are very good, except in one patient who unfortunately died. And the good news, if there is any silver lining in this news, is that the death does not appear to be the result of the gene editing per se. Okay. And the FDA agrees with that conclusion. Otherwise, the trial would have been halted. But rather, it was an adverse event from the chemotherapy. Remember, I said it's a brutal process, even for the really kind of healthy, strong people. And it appears that it was the chemotherapy that really ailed this patient, nothing to do with the DNA editing. So 5 to 10 years, we would hope that we would now potentially have dozens of clinical trials underway, and hopefully 10 20 approved therapies. I also hope that the lessons of CRISPR will help reveal new biology and new approaches that it doesn't really matter at the end of the day whether we cure sickle cell or Alzheimer's disease by gene editing or by a small molecule drug. We just want to help patients to live long, healthy, fulfilling lives. And CRISPR will help reveal a lot of biology in that regard. We'll have to come back for another episode because there are so many other crazy, wacky
Yes.
applications of CRISPR. My favorite is probably George Church, who we mentioned earlier, who has a company called Colossal Biosciences. He's trying to resurrect the woolly mammoth, not to build a
Yes.
theme park, but to hopefully help in to counter global warming. So if you layer the genome of the Asian elephant and the DNA that we've been able to mine from extinct specimens of woolly mammoths in Siberia and many other places, we can line these two genomes side by side, and we can sort of cherry pick key genes that may encode for certain traits. Body hair would be, or animal hair would be one of them, and kind of stitch them into the Asian elephant genome with the long-term plan of potentially breeding a hybrid creature that would look and resemble much more closely the woolly mammoth than the Asian elephant. Why would we do that? Because if we could restore these creatures to the frozen tundra of the plains of Siberia, we'll keep the permafrost tapped down because there are gigatons, I'm guessing, but there's a lot of methane buried just under the surface in these regions. And if that was to leach out into the atmosphere, then life would really become unpleasant. So lots of fun things for CRISPR's future, and or whatever the new version of CRISPR becomes, because even as we speak, researchers are publishing new forms of gene editing that are really exciting.
That's great. Well, we'll have to talk. I would love to talk with you more about this in another episode as well, and we'll see if we can get that lined up. But as we wrap up here in today's episode, once again, I've learned a lot. Thank you so much, and thank you for that wonderful book. People, you will want to look and go get this book. It's a great read. You're going to learn a lot and open your eyes to just what's happened in this space. It's amazing. But before we wrap up, I do want to ask you if you can talk about your CRISPR journal.
Yeah.
So people can go to that to get
you
more
can
information.
www.crisprjournal.com. So when I started my plan to write Editing Humanity in about 2017, I was so obsessed with CRISPR at this point that I thought, there should be a CRISPR journal. Why don't we launch the CRISPR journal? And I started a new job with a wonderful publishing company just outside New York called Marianne Liebert, and brought this idea to them. They loved it. They said, let's do it. And we got this off the ground within a period of about 6 months. I mentioned the chief editor, Rodolphe Barangu. We have a star-studded editorial board, including Doudna, Charpentier, Church, and many other luminaries. And much of the content is for subscribers only. So for any listeners who are part of an organization, a company, or a university, that's really the medium to access most of this material. Mm-hmm. But the editorial I mentioned that Fyodor wrote about how to change the FDA and the regulation of CRISPR therapies to really maximize the potential. We've made that open access, so anybody can read that. And if they can't get a hold of it, just email me, find me online, and I'll gladly send them a copy.
That's great. That's wonderful. Kevin, what else do you have here as we wrap up? Is there any new projects, anything going on?
Well, so we talked about sickle cell, and so you won't be surprised to learn that's my next project. As I wrapped up Editing Humanity, and you read the book, so you'll remember this, there was a fleeting mention to a woman named Victoria Gray, who had been has given several interviews on National Public Radio from the beginning of her therapy. And her results were spectacular, as were the results of everybody else in the trial. The next patient in that trial, Jimmy Oligari, just successfully climbed Mount Kilimanjaro. So that's proof, if you ever needed it, that this is essentially a full cure. And I thought sickle cell is such a famous disease, such a historic disease, but yet such an undertreated, underfunded. There's a whole association with the racism that patients receive, because the main hallmark of sickle cell is it's the anemia in the name, and B, it's the pain crises for which they need the strongest possible drugs, and they're often accused of being drug seekers when they arrive at hospital. So there are many dimensions to this story. I've interviewed a handful of patients who've now been cured. These interviews will live with me. I went to visit Victoria in her hometown in Forest, Mississippi, and we did the interview in the front pew of her church, her local Baptist church, which we were both crying by the end. Me, because I brought her to tears. She, because she was reliving the pain and the emotional episodes of lying doctors, racist nurses. It was pretty shocking to hear such vivid testimony firsthand. So anyway, my next book will be published by Harvard University Press, probably sometime in 2026, and the working title is Curved Air.
Wow. Awesome. I'm looking forward to that as well. Okay. People can follow you the way that, these areas. Is there a website? Is there anything like that?
I'm on LinkedIn. I'm on X. I'm on, a lot of people are now either abandoning or taking a pause from X to try out Blue Sky, which is suddenly since the election, there's been a flood of science communicators and science writers and scientists. So I think for this audience, do look at Blue Sky. I think you'll find some very civilized conversation going on there. So look for me there.
That's great. Well, thank you, Kevin, once again for this. This has been awesome. Let me keep you for 1 or 2 minutes after we stop just to chat.
Sure.
And I think that this is some of the unexpected and amazing breakthroughs in the future and now. So everybody keep watching these things, see where CRISPR goes and what's happening. And just remember, there's a lot of strange and unexpected in our reality. So always remember to keep your eyes, ears, and minds open. And until next time, stay safe.
Thank you for listening to High Strangeness. This was a glance at some of the strange that exists in our reality.